Good morning, and my topic is oral mucous membrane, and I am a postgraduate student from Department of Oral Pathology, Sri Balaji Dental College. Introduction. Oral mucous membrane is the moist lining of the oral cavity. The surface of the oral cavity is a mucous membrane, first of all, and it is a continuous with skin of the lip through the verbalian border. Posteriorly, it is continuous with the mucosa of the pharynx. Classification. Depending on the functional adaptation, it is classified into three types. First one is masticatory mucosa, which covers the gingiva, hard palate, and in lining mucosa, lip, cheek, vestibular, fornix, alveolar mucosa, floor of the mouth and soft palate, specialized mucosa, dorsum of the tongue and buds, taste buds. In this diagram, you can able to see the different types of mucosa covering different parts of the oral cavity. So depending on the keratinization, two types, keratinized and non-keratinized. Under keratinized, masticatory mucosa, that is hard palate and gingiva, and non-keratinized, lips, cheek, alveolar mucosa, and floor of the mouth. Functions: it protects, lubricates, scents, and defends. So four major functions are there. Under which protection, it protects from mechanical trauma during mastication, and lubrication, saliva maintains the moist surface, preventing from drying and cracking, and it helps in mastication, speech, and swallowing. Sensation, receptors that respond to temperature, touch, pain, and taste, initiates reflexes such as gagging, swallowing, and salivation. Defense, it acts as a major barrier for the entry of microorganisms. It is impermeable to bacterial toxins. It secretes antibodies and has an efficient humoral and cell-mediated immunity. Coming to the development part, the epithelium of the oral cavity is derived from both the ectoderm and endoderm, the anterior part of which is lined by the epithelium derived from the ectoderm. Tongue, epiglottis, and pharynx have the epithelium derived from the endoderm. Around 6 weeks, vestibular lamina separates from the primary epithelial band. Degeneration of these cells in the central part of this process leads to the formation of labial and buccal sulcus and the delineation of lips and cheeks from the alveolar mucosa. About 7th week, lingual papillae appear, and around 10 to 12 weeks, circumvallate and foliate papilla appears earlier than the filiform papilla. So this video shows the development of ectoderm, endoderm, and mesoderm. So you can see the amniotic cavity, epiblast, extraembryonic mesoderm. Here the precaudal plate is developed. The cranial end, the caudal end, oropharyngeal membrane, primitive streak, and cloacal membrane. Formation of the definitive endoderm starts here. In this part, what happens? See, the upper part, blue one is the epiblast and the yellow one is the hypoblast. So due to the proliferative cells, these cells are regenerated and the definitive endoderm is formed. And again, formation of the intraembryonic mesoderm occurs, where we can see the other proliferating cells which comes in between the epiblast and the definitive endoderm, just forming the mesoderm. Here comes the end of the slide. Next, about 13 to 20 weeks, differences between keratinized and non-keratinized mucosa becomes apparent. Keratohyalin granules in the keratinized mucosa and region-specific cytokeratins appear. The epithelium remains parakeratinized in masticatory mucosa. Orthokeratinization is seen only after teeth eruption. Langerhans cells and melanocytes also appear during this period. In lamina propria, reticular fibers appear 6 to 8 weeks, followed by collagen fibers, which is 8 to 12 weeks, and the elastic fibers appear by 17 to 20 weeks. There comes the structure of oral mucosa, out of which first the basement membrane, connective tissue, and epithelium. Under epithelium, non-keratinized and keratinized, as I said before, and basement membrane, basal lamina, under which lamina lucida and lamina densa. Connective tissue consists of two main things, lamina propria and submucosa. So this diagram explains the different parts of the oral mucosa, where the topmost layer is epithelium. Then comes lamina propria, submucosal layer. In the connective tissue, we can able to see the glands, nerves, periosteum, bone, arteries, veins, etc. Then comes basement membrane. Interface between the connective tissue and the epithelium is the basement membrane. It is a relatively cell-free zone, which is 1 to 4 mm. Ultrastructurally, it is called basal lamina, but it is not just a membrane. It is a basal complex consisting of lamina and fibers. It is made up of a clear zone, that is lamina lucida, just below the epithelial cells and dark zones, lamina densa, beyond lamina lucida and adjacent connective tissue. This diagram explains the different parts of basal lamina, where the lamina lucida and lamina densa can be seen. Comes the anchoring fibrils, which contains type 7 collagen, forms glue and are inserted into the lamina densa. It consists of type 1 and type 2 collagen fibers runs through these glues. And in this diagram, we can able to see the basal keratinocyte, desmosome, hemidesmosome, and lamina lucida and densa, and the anchoring fibrils can also be seen here. Out of which the anchoring fibrils type 7 collagen can be seen in the uppermost part, and in the deeper part, anchoring black type 4 collagen, which is together known as the reticular lamina. Type 1 and type 2 collagen fibers runs through these loops. Lamina densa contains type 4 collagen coated with heparin sulfate. Lamina lucida is a glycoprotein layer with 20 to 40 nm width, and it contains type 4 collagen, an antigen bound by the antibody KF1. Lamina lucida contains laminin. Functions of type 4 collagen and laminin is it promotes epithelial cell growth. Function of basement membrane. Two major functions. It promotes the differentiation and promotes the peripheral nerve regeneration and prevents metastasis. Next comes lamina propria. It is described as a connective tissue of variable thickness that supports the epithelium. It is divided for descriptive reasons into two parts. They are papillary and reticular. As we saw the previous diagram, we can able to see the reticular lamina propria. Here, the major papillary portion is between the epithelial ridges and the reticular portion. Next, the reticular portion contains immature argyrophilic reticular fibers, and it contains net-like collagen fibers. Usually, reticular zone will be present always rather than papillary zone, because the papillary zone may be absent in certain areas such as alveolar mucosa when the papillae are either very short or lacking. So, interlocking arrangement of the connective tissue papillae and the epithelial ridges and the even finer undulations and projections found at the base of the epithelial cells increases the contact between the lamina propria and epithelium. Ground substances in the lamina propria contains glycoproteins and proteoglycans. Apart from fibroblasts, mast cells and macrophages are also present. Collagen fibers. Here in this lamina propria, we can able to see type 1 and type 3 collagen fibers. Lamina propria may attach to periosteum of the alveolar bone, or it may overlay the submucosa. Then comes submucosa. It consists of connective tissue of varying thickness and density attaching to the mucous membrane. Glands, blood vessels, nerves, and adipose tissues are also present in this layer. Larger veins divides into smaller branches while entering the lamina propria. They can again divide into subepithelial capillary network in the papillae. The veins originating from the capillary network course back along the path taken by the arteries. The blood vessels are accompanied by a rich network of lymph vessels. The sensory nerves of the mucous membrane tend to be more concentrated towards the anterior part of the mouth, that is, rugae, tip of tongue, etc. Specialized receptors for cold, that is, cross-end buds, touch, basinus corpuscles, and nerve-ending pain receptors are also formed. The organized mechanoreceptive corpuscles are present in the mucosae of gingiva, cheek, tongue, and soft palate, and also at the hard palate. Shape of which is elongated or globular, and it is located in the connective tissue papillae. The capsules has several layers of cytoplasmic extensions of perineural cells, and numerous bundles of collagen fibers are noted at the periphery of the corpus. The blood vessels are accompanied by non-myelinated visceral nerve fibers that supply their smooth muscles. So, out of which some points to be remembered at this stage while studying oral mucosa, which is type of covering epithelium is degree of keratinization and thickness of the epithelium, then structure of lamina propria is density and elasticity, form of junction between the epithelium and lamina propria, and the submucosa. So these points must be remembered when studying oral mucosa till this stage. So then comes structure of oral epithelium. Epithelium is stratified squamous type, as we know. Then depending on the location, it may be keratinized, which is ortho or parakeratinized or non-keratinized. Keratinized epithelium, which is seen in the gingiva and the heart palate. Non-keratinized epithelium, which is seen in the cheek, facial and sublingual tissues. Type of cells in the epithelium is either keratinocyte or non-keratinocyte. Cytokeratins. It forms a cytoskeleton of all epithelial cells along with microfilaments and microfibrils. It is seen not only within the cell but also in the contact areas like desmosomes. Functions. It provides mechanical linkages and distributes the forces over a wide area. And it is a stress-bearing structure within the epithelial cell and are important in maintaining the cell shape. So it is otherwise known as intermediate filaments because of the diameter, which is 7 to 11 nm, which is the intermediate between the larger microtubules, that is 25 nm, and the smaller microfilaments, 4 to 6 nm. The molecular weight vary widely from 40 to 200 kDa than microtubules 55 kDa and microfilaments 25 kDa. Cytokeratins is basically divided into two types, which is acidic cytokeratin and the basic cytokeratin. Basic cytokeratin consists of type 1 to 8 and type 2 consists of 9 to 20 at the acidic cytokeratin. Keratinized epithelium. Keratinized epithelium has 4 layers. As we all know, stratum basale, stratum spinosum, stratum granulosum, and stratum corneum. So this diagram explains the different layers of the epithelium. Process of keratinization. So after mitosis, what happens? Keratinocytes remain in the basal layer and divide again and again, or it may become determined, during which time it migrates and pushed upward. Migration. Then comes the migration. Keratinocyte is committed to biochemical or morphological changes. Then differentiation. Differentiation ends with the formation of keratinized squama, a dead cell filled with densely packed protein contained within a toughened cell membrane. Then comes desformation. After reaching the surface, it is shed or cast off. This process of shedding is known as desformation. Then comes the maturation. Process of cell migration from the basal to surface is the maturation. So, what is the time taken for this cell division and what it is called? Time taken for cell division and pass to the entire epithelium, which is known as the turnover time or turnover rate. The turnover time of non-keratinized epithelium is faster than keratinized epithelium. Maturing cells produce growth inhibitors which restrict it from further cell division. During migration, cells increases in size and shape. Increased synthesis of tonofilaments occurs in the keratinized epithelium. In non-keratinized epithelium, tonofilaments are few in number but also not aggregated into bundles. Keratinocyte division is stopped only differentiation occurs. For the tissue to remain in a steady state, undifferentiated cells remain in basal layer and form one differentiated cell from each cell that is formed. So this is the cycle. So how come the last keratinized layer is formed from the basal layer? So first comes the basal layer, then the prickle cell layer. So this is the migration of cell from the basal cell to the gingival sulcus. So prickle cell layer, then comes the granulomyelin granules. Granular cell can be seen, at last pycnotic nucleus can be seen. Then comes stratum basale, made up of a single layer of cuboidal cells which synthesize DNA and undergo mitosis. The basal cells in the parabasal spinous layers are referred to as the stratum germinatum, but only the basal cells can divide. Basal cells shows ribosomes and elements of rough surface endoplasmic reticulum indicative of protein synthesizing activity. It has been proposed that the basal cells are made up of two populations. One population is serrated and heavily packed with tonofilaments, which are adaptations for attachment, and the other is non-serrated and is composed of slowly cycling stem cells. The stem cells give rise to slowly dividing cells which serve to protect the genetic information of the tissue. The serrated basal cells are a single layer of cuboidal or high cuboidal cells that has the protoplasmic processes, that is pedicles, projecting from their basal cells towards the connective tissue. So specialized structures are called the hemidesmosomes are found on the basal layer. The lateral borders of adjacent basal cells are closely opposed to connected by desmosomes. So in this picture, we can able to see the desmosomal connection between the adjacent cells. So in the desmosomes, what are all the parts will be there? It consists of connecting filaments, cytoplasmic plaque, cytoskeletal filaments, intracellular space, and plasma membrane. The basal cells contain tonofilaments which pose toward and in some way are attached to the attachment plaques. Desmosome consists of two principal types of proteins. The transmembrane proteins, the desmogleins and desmocolins, are members of the cadherin family. The desmosomal cadherins are linked to the keratin cytoskeleton via the several cytoplasmic attachment plaque proteins, including desmoplakin, lacoglobin, lacofilins, endoplakin, and periplakin. So in this diagram, we can able to see the adjacent plasma membrane and plaque, transmembrane, cadherin, that is glycoprotein. So what are all we are mentioning in the above point can be seen in this picture. So desmosomal and hemidesmosomal junctions provide mechanical linkages and it's seen amongst all oral epithelial cells. So gap junctions. Next comes gap. What are the three types of junctions we know? They are nothing but tight junction, adherent junction, and gap junction. Gap junctions are low resistance junctions and it allows electrical and chemical communication and are occasionally seen. So in this picture, we can able to see the tight junction, adherent junction, and gap junction. Stratum spinosum. It is made up of irregular polyhedral cells which are larger than the basal cells. On the basis of light microscopy, it appears that the cells are joined by intracellular bridges. Tonofibrils seems to course from cell to cell across these bridges, which can be seen in this diagram. The tonofilaments turns or loop adjacent to the attachment plaques and it is suspected that an agglutinating material joins them to the attachment plaques. Desmosomal attachment plaques contains the polypeptides desmoplakin and placoglobin. Monoclonal antibodies to these polypeptides can be used to detect carcinomas by immunofluorescent microscopy. The intercellular spaces contain glycoprotein, glycosaminoglycans, and fibronectin. The tonofilament network and the desmosomes appear to make up a tensile supporting system for the epithelium. Intercellular spaces of the spinous cells in the keratinizing epithelia are large or distended. Thus, the desmosomes are made more prominent and these cells are given a prickly appearance. So, here we know about the prickly appearance and the reason behind it. Why it is named as a spinous layer then? The spiny appearance of the spinous layer is due to the shrinkage of the cells during tissue preparation, causing them to separate at points where desmosomes do not anchor them together. Of the four layers, the spinous cells are the most active in protein synthesis. These can synthesize additional proteins that differ from those made in the basal cells. Then comes stratum granulosum. This layer contains flatter, wider, and these cells are larger than spinous cells. This layer is named for the basophilic keratohyalin granules, which is blue staining with hematoxylin and leucin. The nuclei shows signs of degeneration and pycnosis. Function: synthesizes protein, but rate of synthesis is different. Tonofilaments are more dense in quantity and are often seen associated with keratohyalin granules. So next comes the calcium and retinoids influence epithelial differentiation by altering the transcription of target genes and by regulating the activity of enzymes such as transglutamines, proteinases, and protein kinases. In this layer, cell surface become more regular and more closely applied to the adjacent surfaces. Lamellar granule, a small granule, small organelle, also known as the keratinosome, oddland body, or membrane coating granule. So while reading across organs, we will be able to see the oddland body, which is nothing but the lamellar granule, which forms in the upper spinous and granular cell layers. The membrane coating granules are glycolipids. Lamellar granules discharge their contents. Then what happens? Intercellular space forming an intercellular lamellar material. So, permeability barrier. This barrier forms the junction of the granular and cornified cell layers. During differentiation, inner unit of the cell membrane thickens, forming the cornified cell envelope. Several proteins contribute to this structure, among which involucrin and loricrin become cross-linked by enzyme transglutaminase. The granules differ in appearance from keratinized and non-keratinized epithelium in being elongated and lamellar in keratinized and circular and amorphous in non-keratinized epithelium. Stratum corneum, made up of keratinized squamae, which are larger and flatter than the granular cells, as we all know. Here, all of the nuclei and other granules, organelles such as ribosomes and mitochondria have disappeared. The layer is acidophilic, which is rich staining with hematoxylin and eosin, and it is histologically amorphous. The keratohyaline granules have disappeared. Ultrastructurally, the cells composed of densely packed filaments developed from the tonofilaments, altered and coated by the basic protein of keratohyaline granules, that is lagrin. Cross-linking of tonofilaments by disulfide bonds facilitates close packing of the filaments and gives the mechanical and chemical resistance to this layer. So in this picture, we can able to see the different stages of keratinization. So from the basal layer, constant cell renewal by proliferation. So next comes the exit from cell cycle, reinforcement of the cytoskeleton, expression of late differentiation markers, lipid extrusion, cornification, which is nothing but the formation of the cornified epithelial envelopes, and deformation, shading of the dead corneocytes. Keratinocyte and non-keratinocyte. These are epidermal or epithelial cell that synthesizes keratin, and its characteristic intermediate filament protein is cytokeratin. They show cell division, undergo maturation, and finally deforming. Keratinocytes increase in volume and in each successive layer from basal to superficial layer. So non-keratinocytes. The epithelium contains a smaller population of cells that do not possess cytokeratin filaments, hence they do not have the ability to keratinize. So they do not show any mitotic activity or undergo maturative changes or deforming. They are not arranged in layers and do not form desmosomal attachment with adjacent keratinocyte. So what happens? These cells migrates into the oral epithelium from neural crest or from bone marrow. Melanocytes, Langer cells, Langerhans cells, and Mucous cells of non-keratinocytes found in the oral epithelium. Out of which, first we'll come across melanocytes, which is derived from the embryological neural crest and migrate into the epithelium. Each melanocyte establishes its contact with about 30 to 40 keratinocytes through their dendritic processes. It appears clear in H&E, and keratinocytes release mediators essential for normal melanocyte function. So it can vary in number in different regions, but variations is the degree of pigmentation, and it is not related to the number, but is due to their aquatic condition. So this is the picture of the melanocyte, where we can able to see the dendrite, melanin, melanosome, nucleus, and the cell organelles, that is area, that is rough endoplasmic reticulum, mitochondria, and Golgi apparatus. Then comes Langerhans cells. So it is found in the upper layers of the skin and mucosal epithelium. There is a correlation in the occurrence of stratum granulosum and Langerhans cells. The cell has a convoluted nucleus and characteristic rod-like granules in the cytoplasm termed Byberg granules. This cell is free of melanin. So in this picture, we can able to see the Byberg granules. It is a cell of hematopoietic origin, and they penetrate the epithelium from lamina propria. It is involved in the immune response. Majorly, it plays an important role in contact hypersensitivity, in anti-tumor immunity, and in graft rejection. Mucous cells. It is found among the basal cells. It has nerve tissue immediately subjacent and is presumed to be a specialized neural pressure-sensitive receptor cell. It responds to touch sensation. They are commonly seen in masticatory mucosa, but are usually absent in lining mucosa. Mucous cells differ from other non-keratinocytes in that they are not dendritic. Ultrastructurally, the nucleus shows a deep invagination and characteristic rod-like. So they migrate from the neural crest and they are stained by pasting, that is periodic acid skip stain. So here the Mucous cells picture can be seen here. So under which sensory neuron and Merkel disc can be seen. So this is just a tabular column which is describing about the melanocyte, Langerhans cell and Merkel cells with some similarities, and it will be easier to remember. So what are non-keratinocytes? Just an animated picture. Then comes the non-keratinization. It is usually the lining mucosa. Basal and prickle cell layers resemble that of keratinized except the prickle cells of non-keratinized epithelium. They are slightly larger and intercellular bridges are less conspicuous. Three layers are seen under non-keratinized epithelium, which is stratum basal, stratum intermediate, and stratum superficial. Basal cells are similar to that of keratinized epithelium, so no need to explain here. Stratum intermedium is there. It is otherwise known as spinous cell layer. In prickle cell layer, increases in size is more than that of keratinized epithelium. Tonofilaments remain dispersed. Certain membrane-bound organelles, they are circular in shape with an amorphous coat. Stratum superficial. Cells of superficial cells are more flattened. It contains dispersed tonofilaments and nuclei. Number of cell organelles are diminished, not dehydrated. They contain less number of tonofilaments and lack keratohyalin granules. So this is a picture where the epithelial maturation in non-keratinized mucosa can be seen. So out of which, what are the points to be remembered? In non-keratinized epithelium, there is no stratum granulosum, nor there is a stratum corneum. This is a very important point to be remembered. Then, tissues that are not keratinized at one stage of development may keratinized at another stage. Tissues may be modulated from keratinized, parakeratinized, and non-keratinized variants in pathologic states. When keratinization occurs in the normally non-keratinized tissue, it is referred to as keratosis. When normally keratinizing tissue, such as epidermis, becomes parakeratinized, it is referred to as parakeratosis. So, what is the difference between the keratinized and the non-keratinized epithelium? In keratinized epithelium, we all know 4 layers can be seen: basal, spinosum, granular, and conifide. In non-keratinized, basal, intermediate, and superficial. It produces a surface conifide layer. It does not produce a conifide layer. Prickly appearance can be seen. Intercellular spaces are not obvious, less or no prickly appearance. Orthokeratinized, no nucleus. Parakeratinized, pycnotic nucleus. Stratum superficial contains nucleated cells. So, in the stratum corneum layer, we can able to see the orthokeratinization or parakeratinization, where orthokeratinization is of no nucleus and parakeratinization is of pycnotic nucleus. Whereas in non-keratinized epithelium, stratum superficial contains nucleated cells. Filaggrin is present in keratinized epithelium and lack of filaggrin but contain involucrin. Numerous tonofilaments, keratohyalin granules can be present in the keratinized epithelium. Less of developed and dispersed tonofilaments, lack of keratohyalin granules can be seen in the non-keratinized epithelium. So this is a picture variation between the keratinized and the non-keratinized epithelium. Subdivisions of oral mucosa. For descriptive purpose, oral mucosa is divided into keratinized area and non-keratinized area. So as we all know, masticatory mucosa comes under the keratinized area. Non-keratinized area, lining mucosa and specialized mucosa. Masticatory mucosa have similarities in thickness and keratinization of epithelium in thickness, density, and firmness of the lamina propria, and is attached to the underlying structure. Heart palate. The mucous membrane of the heart palate is tightly fixed to the underlying periosteum and therefore it is immovable. The color of which is pink. Epithelium and lamina propria we are going to see here. The epithelium is uniform and it is well keratinized, where the cells of the stratum corneum exhibit stacking. The pedicles, the increase in number and length of desmosomes, the density of the tonofilaments, and the complementary grooves and bridges adapts to the keratinizing epithelium to resist moves and to bind the epithelium to the connective tissue. Lamina propria, it is a layer of dense connective tissue which is thicker in the anterior than in the posterior parts of the palate and has numerous long papillae. So this is the histological picture of heart palate, where we can see the epithelium, lamina propria, submucosal layer, etc. Incisive papilla. It is formed of dense connective tissue. Contains the oral parts of the vestigial nasopalatine ducts, blind ducts of varying length lined by simple or superstratified columnar epithelium rich in goblets, and the ducts are surrounded by small irregular islands of hyaline cartilage. Palatine rugae. It is irregular and often asymmetric, extending laterally from the incisive papilla and the interior part of the raphe. Their core is made up of a dense connective tissue layer with fine interwoven fibers. Here we can able to see the dermis, that is lamina propria and palatal rugae. So in the palatal rugae, the HNE stain exhibits the palatal rugae and it is lamina propria as mentioned in this picture. Then comes gingiva. It extends from the dentogingival junction to the alveolar mucosa. The morphology of both epithelium and connective tissue indicates the adaptation of these forces. So epithelium is stratified squamous, may be keratinized or non-keratinized, but most often it is parakeratinized. Then lamina propria. Limitations, it is limited on the outer surface of both jaws by the mucogingival junction. Clinically, it is identified as the junction between the bright pink alveolar mucosa and pale pink gingiva. The color is normally pink but may sometimes have a grayish tint. Divided into three types: free gingiva, attached gingiva, and interdental papilla. So in this we can able to see the attached gingiva, interdental gingiva, mandibular, buccal frenum, mandibular vestibule, and everything can be seen in this picture. So this gives the clinical appearance of gingiva. Okay. Then comes the histology of gingiva. It can be orthokeratinized and thin, long reticles can be seen with connective tissue. So free gingiva. Dividing line between the free gingiva and the attached gingiva is the free gingival group. The free gingival group not always visible microscopically. It appears in the histological sections as a shallow V-shaped notch at a heavy epithelial ridge. The free gingival group develops at the bottom of the gingival sulcus. In the absence of the sulcus, there is no free gingiva. Attached gingiva, it is about 4 to 6 mm in size, and the mucogingival junction is 3 to 5 mm below the level of the crest of the alveolar. Here, the main characteristic is stippling. Stippling is nothing but the portions of the epithelium appears to be elevated, and between the elevation, there are shallow depressions. Disappearance of stippling is an indication of edema and an expression of the involvement of gingiva in the progressive gingivitis condition. Then comes interdental papilla, the shape of which is triangular. When it is tent shaped, oral or vestibular corners are high and central part is like a valley. Depressed part of interdental gingiva is called cone and it is covered by thin non-keratinized papilla. In gingiva, 75% is parakeratinized, 15% is keratinized, 10% is keratinized, and elastic fibers, 10% is non-keratinized. In elastic fibers of lamina propria is known as oxytalin fibers. So here it is divided into following groups of fibers: dentogingival, alveolo-gingival, circular, dentoperiosteal, accessory fibers. So in this picture we can able to see where the dentogingival is extending from the cervical cementum into the lamina propria of the gingiva. The fibers of the gingival ligament constitute the most numerous group of gingival fibers. Alveolar gingival crest of fibers. The fibers arise from the alveolar crest and extend into the lamina propria. Circular fibers, a small group of fibers that circles the tooth and interlace with other fibers. Dentoperiosteal. It is followed from the cementum into the periosteum of the alveolar crest and of the vestibular and oral surfaces of the alveolar bone. Accessory fibers, it extends interproximally between the adjacent teeth and is also referred to as transseptal fibers. These fibers makes up the interdental ligament. Blood and nerve supply, chiefly from the branches of the alveolar arteries. Interdental alveolar artery perforate the alveolar crest in the interdental space and end in the interdental papilla, thus supplying the adjacent areas of buccal and lingual gingiva. In gingiva, these branches anastomose with superficial branches that supply the oral and vestibular mucosa and marginal gingiva. Nerve supply. Different types of nerve endings can be observed here, out of which Meissner or cross corpuscles, end bulbs, loops, or fine fibers that enter the epithelium as ultra-terminal fibers. Lymph vessels is nothing but the submental and submandibular lymph nodes. Verbal lymph zone. transitional zone between the skin of the lip and the mucous membrane of the lip, vermilion border line that separates the skin from the vermilion zone. The skin on the outer surface of the lip is covered by a moderately thick keratinized epithelium with a rather thick stratum corneum. Many sebaceous glands are found in the connection with the hair follicles. Sweat glands also occur between them. Because it is seen in the skin, so we can able to see the sebaceous glands below. The keratinization decreases towards the lip, but the thickness of the epithelium increases. The inner aspect of the lip is thicker and the non-keratinized labial mucosa. So there comes the end of part 1. Meet you in